We're going to take a look at the procedure for doing our egg experiment. And so we're going to have the opportunity to look at diffusion through a semi-permeable membrane as we look at the egg here. This is going to be an extra credit home lab that you can do. So you'll take it about 5 days to do. So essentially it's 5 days for the process itself, and then any time for your lab questions that you have there. And you'll have 2 weeks to complete it. So you can do it anytime within those 2 weeks. Looking at the materials we'll need to do our egg experiment. And so we're going to need 2 eggs, roughly the same size, so that we can see differences within them. We need a glass container of any kind. You can just even use a cup. Doesn't matter what exactly it is, as long as we can put a lid on it, either a plastic wrapped lid, or in this case I have a jar lid as well. We need light corn syrup, or Caro syrup as it sometimes goes by. And so we have Caro syrup in the light corn syrup variety. We need vinegar, preferably white distilled vinegar is going to be the best for this experiment, because we can see through it, we can see through the liquid itself. But you can really use any kind of vinegar that you may have on hand, as long as we can kind of see it through it. It's clear, doesn't have any kind of extra things in it, similar to what you would find in something like apple cider vinegar, where if it's not filtered, you're going to get a whole bunch of different components in there as far as the contents that are there. We need something to write with so that we can mark our eggs. One's going to go into our bottle and be our experimental egg, and the other one is going to be our control egg. For day 1, we're going to take our 2 eggs, and one of them we're going to mark as our control. So we want to put some sort of mark on the egg. This egg, just keep in your refrigerator. That one we want to keep aside. You can keep that in the refrigerator. The other egg is going to be our experimental egg. We're going to place it into the jar or the cup, whichever you happen to have. One of the things to potentially do is to put it in there and then fill it with water just till it covers it. Take that, pour it out into your measuring cup, and then measure how much water you have in there, so that you know how much vinegar you're going to need. Then once we have our vinegar measured out, and I already pre-measured it here, we're going to pour it over the egg so that the egg is covered, and then we're going to lightly cover the jar itself. And so in this case I'm just going to lightly cover it. It doesn't have to be sealed tight. Same thing if you use plastic wrap, just loosely covered over it. And then we're going to let this egg sit. This egg can go back into the refrigerator, because this one's still going to be edible when we're done. This one is not. For day 2, all you need to do is observe your eggs. And so on this day, you're just going to take a look at your egg that is your control and your experimental egg, and compare the two and write down any observations that you see there in the differences between them, and then wait another 24 hours for day 3. When you are writing down your observations, you'll put any observations that you see within the eggs themselves over here or in the liquid, and write those down in that space there. For the extracellular volume here, so essentially we have the egg, which is kind of like a cell, and then that cell has liquid inside it, and then there's also extracellular fluid as well. And so we're measuring initially that amount of extracellular fluid that we started with. And in my case, I believe I started out with about a half a cup of vinegar on day 1. And I'm not going to measure that again until I get to day 3. And I'm going to measure that amount and see what I have on day 3. And so see if we have any loss or gain of fluid there in the vinegar. And so we'll take a look at that. And then when you start out on day 3 with your corn syrup, you'll again look at the amount of fluid that's there. And so if I again used half a cup, then I would write that down there as well, and continuing to write any kind of observations that you see on each day as you go along. Day 3, we're going to again make some observations of our egg in the vinegar, and we're going to take the egg as well, and we're going to measure how much liquid we have left inside the jar. So we need our measuring cup, take our egg, carefully pour it out, get all the liquid into there as much as possible, and then we can again observe the egg, mark any of the components there in your chart. We're going to measure and see our volume that we have left, and that's going to get marked in the chart as well. From there, we're going to take this egg just the way that it is, and we're going to add our corn syrup to it. And so we're going to add that again, similar to what we did for the vinegar. Measure how much vinegar, or use the same amount of vinegar that you had for your corn syrup, measure it into the cup, and then pour your corn syrup back over the egg, and then we're going to let that set for 24 hours. Now that we've had the corn syrup sitting in the egg for a day, we're going to again pour off the corn syrup, try and get all of it out. You may need to scrape it a little bit. And then we're going to take the egg and wash it. I've already pre-washed it and put it back into the container. And then this time we're just going to fill it with water. Again, measure your water first, fill it over to the top of the egg, and then we're going to let this sit for 24 hours once again. We've measured our Caro syrup, put that in your chart, make any observations of the egg itself, and then lightly cover the water container at this point, and now we wait 24 hours one more time. Day 5, we have our egg that's now been covered in water. We're once again going to pour out the water, measure how much water we had left, make our observations of the egg itself, and see where we're at for day 5, and then go ahead and fill out all of your analysis questions for the lab and turn those in. After completing all 5 days of the experiment, now you have your list of lab analysis questions to complete. Again, you can do this in either a Word document or have printed it out and filled it out by hand. If you do the Word document, you really only need to, again, save that second page and send that page along as a Word file into your assignment link. And same thing, if you write it out by hand and you're going to turn it in that way, then just make sure that you print it out, take a photo of your completed assignment, and upload that into Blackboard for your assignment as well. And so just that last page is all that's needed to do. And then I will take a look and see how your experiments came out. And hopefully you had a good time going through the experiment itself and getting a little bit of live lab action.